Thanks, everybody. Thanks for taking the time and joining and meeting us today for our webinar on understanding biologicals in agriculture. My name is Matthew Fletcher. I'm the technical sales representative for Eastern Canada. I'm excited to have the opportunity to introduce our expert lineup of Luluman speakers today. Our first speaker will be Alexa Pierce. She is our innovation project lead who is based out of Saskatoon. And then following Alexa will be Dr. Megan Rains. She's our field solutions manager based out of Sault Ste. Marie. And then to finalize things and wrap things up will be our senior technical sales representative, Chris Zemlak, based out of North Sask. Throughout the presentation and the webinar, if you have any questions, please just submit them via the Q&A tab at the top of your screen. If we cannot answer your questions during the session, we will follow up with an email. So please feel free to ask any questions during. So we'll go to the next slide here. Yep, and so before that, just in case you're not familiar with Luluman as a company, I'll do a quick overview. So Luluman is a privately held Canadian company established over 100 years ago. We solely focus on research and commercial development of microbial technologies. And so as I mentioned, we are a Canadian company headquartered in Montreal. However, we do operate on a global scale with 12 different business units. And these business units there are off to the right. And you can see them, from everything from distilled spirits to brewing, enology, to things like probiotics and animal nutrition. Myself and my colleagues on this call, we all work within plant care division, providing agricultural solutions. So that's a little bit of Luluman. So our next slide here showcases some of the consumer-facing products or everyday uses that Luluman's 12 business units are integrated within. So this just showcases how frequently people might engage with Luluman without even knowing it. So, looking at 1 out of 3 bottles of Scotch whiskey, 7 out of 10 bottles of wine, probiotics, ethanol, silage inoculants, and so on. And so with that quick background on Luluman as a company, we're going to dive into our topic of understanding biologicals in agriculture. And I welcome Alexa Pierce as our first speaker. So Alexa.
Okay. Thank you so much for having me and for being here today. I'm just going to jump right into the material because we have a lot of interesting things to talk about and not a lot of time. So I just wanted to start with something kind of broad. So what is actually a biological? It's actually an umbrella term that can capture many different things. So a few examples here on the screen are things like biochemicals. So this will be our humic acids, things like organic acids or vitamins. And we also have plant extracts that could be considered a biological. So essential oils or kelp or seaweed extract would be a few examples. The next category would be living organisms. This is our beneficial bacteria, fungi, yeast, or insects. And then everything else just kind of gets put into the other category. And this would include animal-derived products, so things like bone meal, for an example. So really the point of this slide is to showcase that biologicals really is an umbrella term. So when we're discussing biologicals amongst ourselves or with other people, it's important to get clear on what it is that we're actually talking about because it can refer to many different things. The focus of our presentation today will be on the living organisms. And so when we talk about organisms, what are we actually referring to? So microorganisms are living things that essentially you need a microscope in order to see. So there's a few examples here on your screen, things like viruses, archaea, algae, bacteria, fungi, and protozoa are all classified as microorganisms. But our focus today is really going to be on the bacteria and fungi. And this is because these are the microorganisms that are mainly used in agriculture. So to talk a little bit about bacteria, these are single-celled organisms. They're often motile, which means that they can move around in their environment. So they might move towards a source of nutrition, or they might move away from a toxic substance, as an example. And they're divided into these 2 broad categories, gram-negative and gram-positive bacteria. And what this means is that there's simply a difference in the way that their cell walls are formed. And that's the photo that you're seeing on the right-hand side of your screen. So there's gram-negative bacteria. And an example of this would be rhizobia. And an example of gram-positive bacteria would be bacillus. And the reason that I'm kind of going through this information is because many gram-positive bacteria have the ability to form spores, and this allows them to survive in harsh environmental conditions. So spores are these really robust structures that are allowing certain types of bacteria, mainly gram-positive bacteria, to survive in these harsher environmental conditions. So think about things such as a really high or low pH, an area where there's not much moisture perhaps, or an area where there's really increased temperature. And so some of these innate differences in bacteria become important when we start thinking about shelf life of a product, or why you might have to handle certain biological products different from one another. Fungi, on the other hand, so they can also be single-celled, and these would be yeasts. They can also be multicellular, so things like mold or mushrooms. They obtain nutrients by excreting enzymes into their environment in order to break down different organic materials, and this is why fungi are such good decomposers. We won't go through the life cycle of fungi in detail, but I wanted to share this image because it nicely highlights a couple of the different structures of fungi that we commonly hear, but we might not know what it's actually referring to. So below the surface of the ground is where we'll find mycelium, and this is the network of branched filaments, and each single filament is termed the hyphae. Once we have a mature mycelial network, they're going to start to form spores. So much like the bacterial spores that we just talked about, they also have a thicker cell wall, and they help to resist different environmental conditions. Before we get too far into this, I wanted to just discuss a little bit about how we actually differentiate different organisms. So essentially we use taxonomy, which is a science that classifies different living organisms into different groups. And this allows us to understand how they're related to one another. So you might remember this hierarchy that you're seeing on the screen from high school biology class. And I want to go through a couple examples today because it's really important for our discussion. So the first example is going to be Homo sapiens, or human beings, and chimpanzees. So you can see that we are related to chimpanzees from the domain level all the way down to the family level. And it's at the genus level where we actually differentiate. So all of us on this call are Homo sapiens, or human beings, maybe with the exception of a couple dogs or cats in the background. And so we're all human beings. We all fall into the same genus and species, but of course there's still some clear differences between us as individuals. So when we think about bacteria and fungi, we actually add in another level of classification. So you can see at the bottom here, strain has been added after species. So in this context, we can say that each individual human being would be a unique strain. And so the example that we'll go through now, which is a bit more relevant to our discussion today, is two bacillus-based biological products that are currently on the market. So on the left-hand side, we have a plant growth promoting product that is also a phosphorus solubilizer. And on the right-hand side, we have a bacillus-based product that is marketed as a biofungicide. So you can see that they are the same from the domain level all the way down to the genus, and it's at the species and strain level where they start to differentiate from one another. And so I'm taking time here and I'm going through this example because the industry as a whole has a tendency to generalize these products and just kind of simplify and say that we have a bacillus product. But that's not the whole story, because a lot of the time the function of these products actually comes from the species and the strain level. So like I said, in this example, we've got a phosphorus solubilizer and a biofungicide. So even though they're both bacillus, they're not the same. So I really just want to get people thinking about this. And think critically about it when we're looking at all the different products that are available on the market. So let's jump into some of the different types of biologicals. We've got three main groups: biostimulants, biofertilizers, and biopesticides. Biostimulants are going to stimulate any natural plant process that improves plant health. This is also a bit of an umbrella term, so biofertilizers will often fall underneath the category of biostimulants. However, biofertilizers tend to be more focused on the actual living microbes rather than different substances like humic acids. So biofertilizers will increase plant absorption capacity, and this is usually through stimulation of root growth and also through making nutrients more available through things like nitrogen fixation. Biopesticides, on the other hand, are the class of biologicals that will offer protection to plants. This is done by repelling, damaging, or killing various pests. Biopesticides is also kind of an umbrella term, and underneath biopesticides, we could have a biofungicide, a bioinsecticide, or a bionematicide. So to get into some examples here, biofertilizers, they can function in a few different ways. So these are just some examples. It's not an exhaustive list. The one we're probably most familiar with is nitrogen fixation, phosphorus solubilization, and then they can also act by increasing water uptake or nutrient uptake. Some examples of some active ingredients that you might see on products that are biofertilizers would be Rhizobium leguminosarum, and this is going to be the rhizobia responsible for nodulating pea, lentil, and faba beans. Another example would be arbuscular mycorrhizal fungi. And this is the fungus that's able to interact and form a symbiotic relationship with many different plant species, and they'll colonize different root systems. These are the images you're seeing on the right-hand side. They'll colonize root systems, and they will start to grow and form that mycelial network that we were talking about. So you can see on the bottom image, those roots have a lot of fine threads and filaments, and that's that mycelial network. And so that's really going to increase the surface area of your root system and allow for more nutrient and water uptake. Another example is Bacillus velezensis, which we will talk a little bit more in detail about later. Jumping to biopesticides, they can have many different functions and mode of actions, and Megan will hone in on this, but a few examples, they might work through producing different toxins, they might have antifungal properties, or they might induce kind of an immune host response in the plant. Some examples of active ingredients here would be Streptomyces species, Trichoderma, or Bacillus thuringiensis. A fun fact here, Bacillus thuringiensis is actually the bacteria that was used, it produces a certain protein that acts as a toxin to different insects. And so when we talk about BT corn or BT cotton, it was this organism that they actually took that protein from. And that's where you see the BT coming from. To move on now and talk about how biologicals actually enhance crop health and yield, I wanted to start with this and pose the question, and it's a question we get all the time: why do we inoculate our soils when there are already billions of microbes present? And the short answer is that not all microbes are created equally, and this leads to variable benefit to the host plant. So I'm going to just take a little bit of time and walk us through this image because I think it's really important for us to understand. This image was shared through a paper, and they were looking at how different rhizobia strains were competitive against one another. So along the bottom axis here, we've got strain competitiveness, and on the left-hand side, we have shoot dry matter, and this is a function of how much the plant is benefiting from the rhizobia. So we have in orange here, we can see that there was strains that had a really nice impact on shoot dry matter or overall plant growth in orange. However, they weren't very competitive, which means that when they were with other bacteria in the soil, they weren't actually able to form nodules on those plants very well, and they got outcompeted. On the blue line here, we had strains that were quite competitive, so they were more able to form nodules on the plants. However, they didn't provide as much benefit to the plant as these orange strains, for example. And what we do as a company that produces inoculants, we're honing in on the bacteria, the strains specifically, that are both competitive and effective. And this is really important, and it answers that question, why do we inoculate? It's because there's many, many strains in the soil already that are going to be looking to associate with the plant because they get benefit from the plant. However, we want to make sure that they're also providing that benefit to the plant. So they need to be both competitive and effective. Like I mentioned, nitrogen fixation is probably the most common example when we think about biofertilization. So this is a process that is it's an enzymatic process that converts nitrogen gas into ammonia. And this is really important because although there's a lot of nitrogen in our atmosphere, about 78% of the air we breathe is actually nitrogen gas, it's in a form that is unavailable for the plant to use. So it needs to undergo this conversion. And that's the equation that you're seeing across the bottom of the screen there. And this is mediated through an enzyme called nitrogenase. So biological nitrogen fixation can happen through symbiotic relationships, like the one with rhizobia and legumes, or it can also occur through asymbiotic relationships. There's free living bacteria that don't need to associate with plants that are able to do this in the soil. And it's a really important process. It contributes about 140 million tons of nitrogen annually. So we're not going to go into detail about this process because it's actually quite complex and it'll take a bit of time, but generally legumes will release root exudates into the soil, and this is going to recruit specific rhizobia. So the two examples on the screen here, soybeans and peas, they are going to release different exudates, and it's going to be specific to the bacteria that will help them form nodules and fix nitrogen. So then the rhizobia are actually going to be able to induce nodule formation, and this creates an environment that is limited in oxygen, which is the key to nitrogen fixation. So if any of you guys on the call here are actually out in the field and pulling up plants and looking at nodules, I would recommend that you also go kind of one step further and start opening up those nodules if you're not doing that already. And so it's really important to be able to see this reddish pink color in your nodules. If you're not seeing it, that means you're not actually getting active nitrogen fixation. And so, yeah, I would just really recommend that you guys take it one step further and check to see whether you're getting that pinkish color. Oftentimes you'll see nodules that are kind of white, greenish, brown. I've even seen black ones before. That's going to mean you're not actually getting nitrogen fixation. And it could be a strain that was maybe highly competitive, but not very effective, that is nodulating that plant. To switch gears now to talk a little bit about phosphorus. Of course, it's another really important nutrient for plants. It is often there's usually a lot of phosphorus unavailable in soils. There's lots of phosphorus in general, but most of it is in a form that is bound up. So we've got kind of two major pools, organic and mineral phosphorus. What actually happens is that these two pools are often bound up by organic material or other cations in the soil like calcium, iron, or aluminum, which means that there's only a small proportion of phosphorus in your soil that is available to the plant to use. And even a smaller amount of that phosphorus is actually reachable by the plant roots. And so this is a problem, of course, because phosphorus is required for many different plant functions, including root growth, maturity, and of course, all those things lead to overall yield. Phosphorus, it depends on your soil, of course. Everyone's soil types are going to be a little bit different, but up to 90% of applied phosphate fertilizer can become unavailable even the same year that it's applied. And like I mentioned, it gets bound up by other molecules in the soil. And so to talk a little bit about mineral phosphorus, what this looks like, it's really a question of pH. So when we're at a pH of about 7 or just a little bit lower, that's when you're going to have the highest phosphorus availability just based on chemistry alone. If we are higher than that or lower than that, that's when you're going to get some of those other cations really binding to that phosphorus and making it unavailable. So here's a couple different examples of how one bacteria, Bacillus velezensis, can make phosphorus available to the plants. So this bacteria works on both the organic and mineral pools of phosphorus in the soil. So organic phosphorus, a few examples of what this is going to be soil organic matter, different manure, maybe plant biomass, other microorganisms in the soil. Again, depending on your soil type, 30 to 90% of phosphorus is going to be in the organic form. And so how Bacillus velazensis is going to make this available is through the production of an enzyme called phytase. And this causes the mineralization of phytate and a release of the organic materials. So on the left-hand side, the image that you're seeing there is Bacillus velazensis on an agar dish, a Petri plate in the lab, and this is just used as a demonstration. And the halo around the colony is an indication that those bonds are being broken and the phosphate is being released. On the other side here, for mineral phosphorus, the major mechanism for mineral phosphate solubilization is through the production of organic acids. And so the example here, we have another Petri plate. It contains a pH indicating dye. And so as the pH starts to drop, you'll see the plate change color. So I'll walk you through this. I'll start playing the video. So on day one, we've added our Bacillus velazensis here. You can see they're producing organic acids. We're getting a drop in pH on this plate. You can now more clearly see the growth of Bacillus velazensis, and they're continuing to produce organic acids. It's a little bit hard to see in this video, but you're also getting a halo forming around that colony, which is indicating that the bond between, in this case, calcium and phosphate has been broken, and therefore the phosphorus is available now for the plant to use. And you can see how much that pH has changed in the course of only 4 days. I'm going to hand it over to Megan Rains now to talk a little bit more in detail about the biopesticides.
So hi, everyone. Thanks, Alexa, for that overview of biofertilizers. So as she mentioned, I'm here to talk today a bit more about biopesticides, and then we'll wrap up with some considerations and challenges of things that you might want to think about if planning to incorporate biologicals into your growth programs. So we know that healthy soils really are going to have a good balance of chemical, physical, and biological properties. We heard Alexa talk about pH dependency for phosphorus. We can also think about the physical attributes of soil, things like soil structure or the tilth, and just the general makeup of your soil, whether it's clay or a sandy loam, for example. But one concept of soil health that's usually pretty important is the biological or the living portion of your soil. And when we have a healthy soil, what we can actually generate is pathogen suppressive soils by the microorganism communities that exist there. So when you have a healthy kind of thriving environment for these microorganisms, they can actually help to suppress diseases such as Pythium, Phytophthora, Rhizoctonia, for example, by both direct and indirect mechanisms. So these healthy soils tend to contain large proportions of beneficial organisms such as Pseudomonas, Bacilli, Trichodermas, or Streptomyces. And they'll actually help to improve your yield by suppressing those diseases that would ultimately lead to plant die-off and rots, for example. So they work by a couple different models. Plants tend to work through something called the cry for help model. And what'll actually happen is when a pathogen attacks a plant, the plant will put out change in its root exudates. And those exudates will actually help to recruit beneficial microorganisms or beneficial bacteria and fungi. And when the pathogen's there and the plant puts out that sort of call for help to the microorganisms that are in the surrounding soil, you'll actually get an enrichment of these beneficial organisms in that root zone, and they'll be working to help suppress disease. Unfortunately, in agriculture, we tend to harvest our crops on a yearly fashion, meaning that the subsequent generations of the plants don't get that beneficial microbe defense that the plant has worked hard to recruit. So when we're introducing biologicals, it's really important that we treat crops in a preventative fashion, meaning that we want to get in before the pathogens attack so that we can actually mimic this call for help. So when we apply a biofungicide, what we're typically doing is we're applying a product that'll work with one of, usually two or more actually, of these modes of action. So these are different than your typical chemical modes of action. These are going to be things like antagonistic metabolites or secondary metabolite production, so they will inhibit the growth, or microbes will compete for nutrients or space and actually exclude out the pathogen. Certain microbes can actually feed upon pathogens, or we can have induced host resistance. So if we look at an example here, Streptomyces in this case, so shown on the right-hand side is a lettuce root that's been colonized by Streptomyces K61. We can see that Streptomyces functions by 3 different modes of action. So it will produce secondary metabolites, it will compete with the pathogen for space and nutrients, and it will actually feed upon certain pathogens. So 3 modes of action here. We can see it being effective against Rhizoctonia, Alternaria, Fusarium, and Phytophthora, and Pythium. So if we look at secondary metabolites kind of as an example and what this looks like for a biological, we can see that on the plate on the left-hand side, we have Rhizoctonia allowed to grow freely. And on the right-hand side, we can see the same plate, but now with Streptomyces K61 applied and a zone of inhibition between the two. So we can clearly see that the production of these bioactive compounds or secondary metabolites are able to inhibit the growth of Rhizoctonia. So the pathogen is not able to grow in the presence of Streptomyces, and this is a result of the antagonistic metabolites that are produced. So there are some benefits to using biofungicides and incorporating biofungicides. One of those is that there's usually multiple modes of action, and that those modes of action typically exist in different groups than chemicals. So you can use this to lengthen out any sort of resistance that may develop and just kind of vary your program. But one thing that we want to make sure is that prevention is key, and all biologicals are going to be preventative rather than curative. And also they can work in combination with your synthetic fungicides. Just to wrap this up with some considerations that we might want to be aware of, we are kind of dealing with a different scale when we talk about biologicals. So microorganisms are not measured in pounds or tons or any sort of weight measurement. They're measured on a log scale, but it's known as a colony forming unit. And the concentrations of a colony forming unit are going to be on a log scale. So it's a 10-fold scale. So what this means is that small changes, so we can see an example of 10 to the 8 versus 10 to the 10, are going to lead to very large increases. So the difference of 10 to the 8 versus 10 to the 10 is actually a 9,900% increase. So if you think about seconds, we're talking 3.17 years versus 317 years. So be aware of the concentration of the microbes contained within the formulation. On that note, formulation is the second consideration to really think about. Not all formulations are going to be the same. You want to make sure you're choosing a formulation that's right for your equipment and right for what you're trying to achieve. So consider things like liquids versus granular versus peat. The formulation is going to make a big difference, and microbes are going to be happier existing in certain formulations rather than others. Kind of the last key to success when incorporating biological is really considering your environmental conditions and your actual needs. Alexa touched on that not all microbes are created equally. We need to be aware that the right product for the right needs is something we always want to consider, and making sure that we're handling these products in a favorable way. Remembering that microorganisms are not synthetic molecules. Sometimes they need a gentler hand. We want to consider compatibility and things like that. Just kind of treat them a little bit more gently when possible. So the 3 takeaways from this webinar that we hope you have achieved is that microbes have a lot to offer. Specifically, we covered bionutrition and biopesticides. The function will vary significantly between organisms, and there's no one-size-fits-all approach. We need to understand product and formulation in order to best optimize its use. So with that, I think Chris is coming in to kind of wrap up our product portfolio, and thank you.
So I hope this has helped everyone who's attended the call today have a bit more of an understanding of microbials and where they fit within our environment here in Canada. I know this marketplace is getting more and more busy as more companies come to play. And just remember to understand some of these key points that we talked about, and that not all products, even with our own portfolio, looking at it globally, will fit within the Canadian marketplace. So looking at the next slide, we have a number of products that we have brought into the marketplace over the last number of years for specific reasons. And ultimately, a lot of that comes down to our unique environmental conditions that we deal with here.
To reiterate the thanks from my colleagues here with Lallemand Plant Care, I'd like to thank everybody for joining our webinar. And if you do have any questions, please feel free to contact your local rep, email our support line, or give us a call. We'll be happy to follow up with those questions later on. Appreciate everybody for attending. Thank you very much.